in biochemistry, because chapter 1 was really a review of biology, chapter 2 is a review more of Gen Chem. Remember, don't forget to finish on your own Henderson-Hasselbalch issue, and that's a little bit of the PowerPoints from chapter 2. Okay, now we're going to actually start with our first biomolecules. So we're going to start learning about amino acids and peptides, because they are used to make larger biomolecules. Amino acids come together and form to make proteins. Okay. There's a term called polypeptide, which gets used for a couple different things. Polypeptide really means multiple amino acids connected together, but it can also mean a single protein chain. Okay. And so just be aware. Okay. This may be a review for some of you who've taken some of the special topics class, or it may be a little bit of a review for that and then in more detail as well, because this is where we start to really go over each of the 20 naturally occurring amino acids. And remember, you need to know all 20 of the naturally occurring amino acids, their one-letter code, three-letter code, and their names and structures. Okay. All amino acids have the same background, okay? And I don't know why whatever it is, didn't come through, the whole bullet point didn't. But every amino acid has, first of all, the ones that are naturally occurring are called alpha amino acids. From organic chemistry, what does that alpha mean? Because there are beta amino acids. In next semester, we actually talk about 1 or 2 beta amino acids. But what are alpha amino acids? Beta amino acids just are not used to build up and make proteins typically.
Next to the alpha carbon.
Well, it's next to the alpha carbon. It's attached to the alpha carbon. So whatever it is, the amino group is attached. And what does alpha mean? First, or is adjacent to what? Well, it's just a locant of sorts. And so whenever I taught organic chemistry, I would call usually we're talking about whatever carbon, it means one away from, okay? And so whatever carbon gets the primary, it gets the, what's the word I'm looking for? Not primary, but it gets the top priority, which for amino acids is going to be the carboxylic acid. And so I call it the ground zero carbon. So this carbon right here would be the ground zero carbon. The carbon next to it would be the C alpha. One carbon further would be beta, and then it goes according to Greek alphabet, delta, epsilon, and so on and so forth. Okay, and so in the end, it doesn't even have to be a carbon. Sometimes they may say something being alpha, like a nitrogen being alpha, or an amino group being alpha. What that literally means is it means adjacent to. These are alpha amino acids because the amino group is hanging off of C alpha. Okay. They only differ in that R group. Okay. What's the fourth thing that's hanging off of every C alpha? Remember, how many bonds does a carbon like to make? 4. So what's that fourth thing? Because if you notice that carbon in the middle there, there's only 3 bonds there. So what's missing? Hydrogen. So there's a hydrogen that's always implied. Okay. Amino acids in vivo typically are in their Zwitter ionic form. This is the same thing. It's just that they've swapped the orientation here, meaning that the pKa of the carboxylic acid causes it to become a carboxylate, and the pKa of the nitrogen causes it to become an ammonium. Okay. But inside a protein, you can have internal pKa's that differ. So they may or may not have the hydrogens attached to them. Okay. So usually, or many times you see them written in what's called the unionized form, not unionized. So no, it's unionized. But at physiological pH, typically it's going to look like a Zwitter ion. There are different ways to write it out. I'm not going to go, you should know all this, balls and sticks and space filling and the perspective drawings and the little, like the way that I just drew it before, where that's the way that you you should probably, hopefully, feel the most comfortable in writing it that way because of the fact that that's the way it's written typically throughout the book. Okay. Now, anytime an amino acid has something special, know it. Like if it's something that makes it unique, then that's important. You know, God made us that way. There's a reason why. And I say that because out of the 20 naturally occurring amino acids that are used to make proteins, 19 of them are chiral. That means how many are achiral? One. And Three Dog Night says, one is the lonely summer. Little theme song there.
Which one is it?
What?
Which one is
Which one is it? Does anyone know?
it?
It's glycine. And why is glycine achiral? How can something be achiral? Or maybe let's start the other way. How can a carbon be chiral? What does it have to have? 4 different things. And the whole idea is because, you know, technically it's because it can't be, it's got to be, the mirror image cannot be superimposable upon itself. Okay. So therefore, why is glycine achiral? What makes it special? It's R group as a hydrogen, so there are 2 hydrogens in the opposite alpha. It's the smallest, which makes it special. It's actually very important. So, but we're going to fix that in a moment. Okay, so yeah, with the exception of glycine, all of them are chiral. There are a couple that even have 2 chiral carbons in them, so that makes them special. I guess. This is an example of alanine. Of course, you should know what a Fisher projection is. Alanine, the R group is a methyl group. So alanine is the second most smallest. And then they talk about the stereo. Remember, in biochemistry, organic likes to use R and S, but biology and biochemistry typically use D and L, which stands for dextrorotary and levorotary. And literally goes back to the idea dextro means right, levo means left. So the way it rotated light. So all of the, and that's why don't be surprised if you see a D alanine, L alanine instead of R and S. Okay, most that are naturally occurring are L amino acids, and they get their configuration based off of the next one, based off of glyceraldehyde. Okay, which glyceraldehyde, if you don't know what background that you have, I don't know how they go about it in biology and genetics, but it's the simplest sugar. It's a 3-carbon containing sugar. You're going to learn more about it later on. Okay. And so that's why it's based off of it. And they always look at the penultimate carbon with respect to the sugars. But once again, that's a different chapter. Okay. All right. And one time I remember I went to a lecture and they were, as a theoretical biologist or biochemist, whatever, they were trying to discuss about the reasons why, they preferred L versus D. Why, evolutionarily speaking, why was that the conserved one versus, so, and there are some exceptions, but. And one thing is more enzymes that recognize amino acids, typically they tend to recognize the L form versus the D form. Just an FYI. All right. So now we get to the 20 naturally occurring amino acids. I've included this here, but I'll tell you right now, I don't like the way that they did it. I find it kind of confusing. So I'll probably just draw these out myself, or draw out the R groups at least. Because remember, they all have the same backbone, but the R groups change. So what this figure here is trying to show you is the R group. Now, that being said, and it gives you like their 1-letter and 3-letter codes, where it gets confusing is with the exception of glycine. I'm trying to think if there's any other exceptions. No, with the exception of glycine, Every R group has at least one carbon hanging off of it, and there's always a linking carbon. Well, the methyl group, there's only one carbon. I say that because if you look here at phenylalanine, this looks like a benzene ring. But in reality, it's a CH2 group and then the benzene ring. That's why it's called phenylalanine, because it looks like alanine, which is a methyl group, and it's got a phenyl group attached to it. And so the way that they have it drawn here, it looks like it's just a benzene ring. Okay, but technically, it has that methylene carbon and then the benzene ring. Just like tryptophan, that's a linking carbon and then the tryptophan. Okay, and proline, we'll get to proline. That's just screwed up looking. Okay, and that's why I'll explain proline in a moment. So that's here for you as a tool to have, but I think your book does a much better job in table, I think it's table 3.1. It's in chapter 3. Okay. All right, so that being said, whoops. Let's just go make it small. Let's go through, take some time. So now we're talking about the ones that are nonpolar. In particular, a lot of times they're called aliphatic. But these are the nonpolar. Now, that being said, there are a couple amino acids that can fit in more than one category. Sometimes methionine, like this list, is added into the aliphatic and nonpolars. Sometimes, because there is a difference between sulfur and carbons, sometimes methionine is added into the polar group. It's just that it's not very polar. And so that's why that one's kind of a gray. So looking upon which book you use, they may have methionine in this group, and they may have methionine in another group. Okay? Just like tyrosine sometimes is located to a similar distribution as well in a moment. But okay, remember, every single amino acid looks like this, for the most part. All right, draw it as this order ionic form. That's the C alpha. We have the amino group. And then we have R. So now we're going to be drawing just the R groups, because otherwise it's going to be a pain. If you want to, you can always draw it out every single time. The simplest. So let's start with, in the beginning, no. It's glycine. Glycine's 3-letter code is easy, GLY. Once again, one thing I don't like about that table is, technically speaking, on technicalities, that first letter is supposed to be a capital, and the next two are supposed to be lowercase. And the reason why is you have to be able to differentiate between the 3-letter code and 1-letter code. The 1-letter code for glycine is easy as well. It's G. Its R group is just a hydrogen. One thing that makes it special is the fact that it is the only one that's achiral. Why do you suppose that's important? Why does it matter that it just has a hydrogen? Think about it.
Small.
It's small, so what can it do? Did anybody ever watch Ocean's 11? At least 3, maybe George Clooney. Okay. Well, there's at least a part, I think it's in Ocean's 11, Ocean's 12, it doesn't really matter. They're all the same ilk, right? But there's the scene where they sneak like a box in, and it's got the small Asian man inside it, right? Because he can fit, okay? That's glycine. Okay, there are times inside the protein where you need to make a sharp turn, or you need to fill in something that's hydrophobic. That can make great turns. Many times it's found in loops, because it only has a hydrogen. So that's why it can make really sharp turns. It's not bulky, it can fit in tight places. Okay? And so that's why it is useful. Okay, the next one, just one up, is alanine. Alanine is ALA, like Alabama, as in Sweet Home. Oh, I used to try to do like theme songs and stuff for every lecture, but it became kind of tiresome between classes to upload it. If so, it probably would have been Sweet Home Alabama for today. And then its 1-letter code is A. It's pretty simple. Its R group is a methyl. So it too can be found in loops and found in tight places. We're going to find out alanine next semester is also very important. Does anyone know what else it's important for? It's very important for something. Does anyone know? I mean, that would be like worth bonus points if you know it right now. But alanine is an amino acid. Okay. There are other types of alpha acids called alpha keto acids. Okay, where instead of it's the ketone instead of the amino group. It's the alpha. So, does anyone know what the alpha keto acid is of alanine? Alanine is a natural source for a very important compound, and that compound is pyruvate. So you're going to find that alanine and pyruvate. If you need energy, you can actually utilize alanine to help you get energy because it's one of the ways that you make that proteins can help you get energy. Because alanine can be converted to pyruvate. Pyruvate can go on for the citric acid cycle, citric acid, so on and so forth. Alanine, pyruvate is also used for it's involved in other metabolic pathways. And so alanine is always a way to get to pyruvate. So that's just the whole. Has nothing to do right now with proteins, but that's why you're going to see alanine a lot next semester. All right. Then we have valine. Valine, its three-letter code is V. Oh, whoops! I already gave it to you. It's VAL. Valine. It's VAL, and its one-letter code is V. So if this is the C alpha, so that's the backbone of the C alpha. There's a reason why I drew it that way. Remember, every single one except for glycine has to have at least one carbon here. Valine, if you draw it this way, looks like this. So if we turn it, oh, I forgot that doesn't work there. So if everybody does this, and you turn, or imagine doing this, it's a V. For valine. I should have, I forgot. I'm trying to just retain my iPad, but it's locked. So it lost a little something in translation there. But it's a V, okay? It's a V for valine. It's hydrophobic. It's called a branched amino acid, so it's got a branch in it. And so for anyone who works out and stuff, you know you have to have the BCAAs, which stand for branched chain amino acids. And valine is one of those. And so valine, isoleucine, lysine. All right. So next up is lysine, which is also a branched chain amino acid. It's LEU, and it has a one-letter code of L. So there's the backbone. It has to have at least one linking carbon. Okay. And what it does is then it's the V. So it's the same as valine, but it has an extra linking carbon. So I've seen people get them mixed up. That's why the number of carbons in CH2, which really doesn't matter. Now, sometimes you may have you heard of the term like especially in genetics when they start looking at point mutations and they say that there's something that's a homologous or a conserved point mutation, something that doesn't, or like a conservative mutation. Valine and lysine many times can do that. Since they're both hydrophobic, they have similar shapes. It's just that since lysine is bigger, sometimes it can cause some structural issues, or that valine is smaller, it maybe it's not as, maybe there's some extra space in there, which we need that. But otherwise, usually, if you have a point mutation for an enzyme or protein of a B to an L or an L to a B, usually it's not life and death, assuming that it doesn't do some gross structural damage. Alrighty. So next, whoops, sorry. Yay. So once again, this is the backbone. It's isoleucine. Isoleucine is one of my least favorites. I'll give it the one-letter code first. It's easy. It's I. The reason why, at least whenever I was memorizing amino acids, I did not like isoleucine because its three-letter code didn't make sense. It's not iso. It's ILE, which, and people in biochemistry a lot of times, maybe they form like a bond to these amino acids, and so they refer to them by their like three-letter codes and things. So they'll say like gly and alanine. So whenever I took a structural biochemistry class and they would talk about it, it told me that one woman would end up saying isoleucine, or calling it IL or ILE. She always said ILE. That three-letter code. So, but yeah. No, I just don't. I'm not a big isoleucine fan. So what do you suppose, if you just knew the name, isoleucine, what does it sound a lot like? Leucine. Mm-hmm. And iso, what do you suppose where does the iso come from? It's an isomer. Okay? And so you know what leucine looked like. Leucine looked just like valine, but it had that linking carbon. Well, isoleucine is very similar. So they all have to have that linking carbon. But remember, this was, don't draw this part. This was leucine, right? Isoleucine just moves that other one up one carbon. So isoleucine gave me fits. I'm still bitter. Okay. Okay. So the next one I'm going to talk about is proline. So I'm going to kind of skip it, the order that they're on that slide. This is proline. Proline is one of my favorites. Proline's one-letter code is P, and three-letter code, pro. It's one of my favorites because it makes perfectly logical sense. Now, where it gets tricky is in its structure. All right, so this is the way that I would draw it. This is one where I draw out the backbone. Okay, so this is what, the backbone would look like for amino acids. Proline, why it's special, or one reason why it's special, is its R group is the only one that doubles back and connects the backbone in two places. So it's cyclical. The way that I remember it, that's why I always draw it this way, because this is how I remember it, is it has 1, 2, 3 carbons and is bound back to that nitrogen. That's not a double bond, that's a single bond. Now, depending upon the pH and the pKa, there may be 1 or 2 hydrogens hanging off that nitrogen. This is why if you look on a lot of the books, it'll have the 2 hydrogens showing the Zwitter ionic form. Sometimes they'll show it with just having 1 hydrogen. It just depends upon whether or not the proline is already in a peptide bond and also what the pH, the local pKa and pH is. Okay. So, what do you think is so special? What's something special about proline? It's a ring. So, what is it aromatic? No, it is not aromatic. So it doesn't have that special, aromaticity. I don't know how much, Dr. Southport went in. So that special property. So what else makes it special? It's a ring. What is its limitation?
It's rigid.
Pardon?
Rigid.
It's rigid. So if you think about even valine, leucine, isoleucine, especially glycine and alanine, they have flexibility. They can move around C alpha. Proline cannot. It is stuck. Okay, and so because of that, it's really important structurally. Now there's an enzyme that's called racemase that can come in and it can switch it from a D to an L or an L to a D. But you literally have to break open the ring and then reform it. And so because of that, it is rigid. It's kinked. It's a kinky one. It's a kinky amino acid. So it's used a lot of times for structural purposes. There's one protein in particular that about 1/3 of all of its amino acids are prolines. Okay, in fact, there's diseases associated with not having enough. In fact, what they do, almost many of them in that protein, they add an alcohol to it. There's another enzyme that comes in and adds an alcohol group, so it can do hydrogen bonding, which is important for this protein. And there are diseases associated with either not having the alcohol, enough alcohol there, or not having enough proteins. Does anyone know what that protein is? It's very important medically. It's collagen. What does collagen do? Connective tissue, skin, bone. And I like to use skin. So, as an example, it's about 1/3 of its amino acids are proline. So if you look at its structure, instead of a normal alpha helical kind of picture, which we'll see those, and you'll look at it's really narrow, kinky, because one of every three is a proline, so it's kinked. And because of that, they literally take one of those strands, and they will take another one and wrap it around it, and a third one and wraps it around it like a rope. So it makes it very, very strong. And between those strands, the hydroxyprolines and hydroxylysine, which we'll get to in a moment, can hydrogen bond to make those strands even tighter and stronger. Okay, so if you don't have enough prolines, or you're missing some of those alcohol groups, then your skin in particular doesn't have the elasticity. Okay, so it's not kinky enough for the collagen, and it also can't hydrogen bond correctly. And so therefore, skin can slag, can slip, and things like that. And so people with that disease, these collagenase diseases, this isn't very politically correct, but it's true. They kind of look like overripe tomatoes. Like, you know, they lose the elasticity. So one of my friends, actually, her coworker had a problem. So like, if you hit something, like his skin did not bounce back, kind of like something tiny or something. All right. Let me see.
So like when people smoke, is that what they're affecting, causing like the dripping?
It can break some of the collagen down. Now, I don't think it's because of the fact that it's rid of proline.
Mm-hmm.
It may just be that it causes breaks in the amino acid chain, or it may cause breaks between the hydrogen bonding, the possibility. But if I had a hypothesis, I don't know, I haven't researched it, is that I would probably say it probably starts to break down some of the peptide bonding there. So therefore, instead of being strong ropes, it's broken, it's fragmented.
But it's not very cool.
Mm-hmm. Okay, so the next one I'm going to talk about, like I said, it kind of belongs in this, but it kind of doesn't. It's methionine, okay? This is methionine. Like I said, some books put this in the polar amino acid. It's an MET for its 3-letter code and M for its 1-letter code. And once I draw the R group, so this is the R group, so here would be the backbone. You always have to have that linking carbon, one, except for glycine, 1, 2, okay. Then there's a sulfur, and then there's another CH3 group. And so the reason why is it's predominantly hydrophobic. However, that sulfur does have a slight dipole with those carbons, so that's why some books then put it over into the polar. I would take it either way. You know, if I asked you to give it, you know, to categorize it, I would take it as either. Okay. Now we have the aromatics. And the simplest of the aromatics, whoops, I hit the wrong one, is phenylalanine. Phenylalanine has a 3-letter code of PHE, so it's Phe. What do you suppose its 1-letter code is? Phenylalanine.
F.
It's F. It cannot be P because that's proline, so they go with phonetics on it. It's F. Okay, so there's the backbone. It has its linking carbon, and then it's a benzene ring. That's why it's aromatic. It is hydrophobic, so that's why it can kind of go in, it goes into 2 groups. It's a hydrophobic one, but it's aromatic. All right. I'm now really skipping on the slides because I wanted to put this one in because it's also aromatic. But it belongs into multiple groups, and that's tyrosine. Okay, so tyrosine, its 3-letter code is TYR. What do you suppose the 1-letter code is for tyrosine? It is not a T. We're going to find out T is 3. So it is Y. Okay. Why? Because I love you. I don't know. So that kind of thing. But no, it's a Y. It went with the 2nd letter. The reason why I put it here is it is aromatic. Okay. So that's why it makes sense. Plus, if you learn phenylalanine, it's easy to learn tyrosine because it's identical with one major exception. Does anyone know what's that one exception for tyrosine? It has an alcohol. And the alcohol is in the P position. If you think back to organic chemistry, it's a 1,4 displacement. And so that's why, yes, it's aromatic. However, it can hydrogen bond. And if you think back to the special properties of phenol, which you guys should cover phenols, right, in organic chemistry? I believe there's usually a whole chapter on it. Is it can easily form what's called a phenoxide, which is where it loses that hydrogen. So many times they also stick this one in the acidic. There are certain ones that are acids. Tyrosine can lose that hydrogen really easily to become a phenoxide. So that's why it can also be in an acid. What else do you suppose tyrosine can do? What are alcohols good for? What's one thing that alcohol's good for besides hydrogen bonding? I already mentioned that one. It could be a leaving group. What else besides a leaving group? It's a good nucleophile. So enzymes sometimes use it. They take away that hydrogen to make a phenoxide, and they can use that as a good nucleophile. Okay. So it's nucleophilic as well. So these are the aromatics. The next one that's an aromatic was always one of my favorites. It's a hard one to draw, but it was one of my favorites, and I think it's just because it made me think of sports. So that is, and it's very important too. That's to do that. It's tryptophan. Oh, don't do that. Fortunately, this is not left-handed friendly. Tryptophan's 3-letter code is not tri. Okay, that one teacher would always call it trip, TRP. And it's 1-letter code. It's just one you have to memorize because it can't be T because T's already taken. It can't be R because R is taken. It can't be Y because Y is taken. It can't be P because P is taken. O doesn't exist. Well, technically O is ornithine, but it's not used to that. So, I mean, you go through it, it's W. Okay, it's just W. All right, so the way that I would do it is you have the backbone. Remember, there's always that linking carbon. That's the one that the mistake that people usually make is they forget that linking carbon. And then it looks like this. And I'll explain how I would, like I said, if you're, maybe it can help you with, if you're in sports, that's a nitrogen. A benzene ring. So, to me, this pentyl heterocyclic group, so there's 5 rings, looks like home plate in baseball with a nitrogen at the apex. It's got a double bond along the side and it's attached to a benzene ring. So it kind of reminds me of baseball. And I guess you can kind of see a W if you look at it really hard right here. Whatever helps you remember. I don't know. But, yeah, it's the linking carbon. Then you have a 5-membered ring with the nitrogen at the bottom, a double bond along the side, and then that benzene ring is conveniently attached.
Where you've always wanted W whenever you're playing baseball.
Yes, true. And unfortunately, it's not the only one that looks like home plate. So we have another one coming out. Okay. All right. So, does anyone know what's tryptophan? What's one of the things that makes it special? What?
Is it like kind of food?
It is in foods, right? With proteins.
Is
What?
it turkey?
Make you. It's notorious for being in turkey, which is not like it's more been in turkey than anything else. It's just because of the fact that Americans eat so much Thanksgiving, I think. But it does make you. It can make you sleepy because tryptophan and tyrosine, for that matter, both are precursors for neurotransmitters. So this one is the one for tryptophan does You know, you get your dopaminergic hormones come from those, or neurotransmitters, I should say, from those. So, yes, it can. You can get like your serotonins and melatonin and all that kind of stuff. Let's see. The last of the aromatics, which is also very, very special, and another one of my favorites. Let me shift it up. It is not hydrophobic. It's actually hydrophilic. And so it's many times in multiple places, and that's histidine. So sometimes it's included in aromatics. Many times it's included as a base and as a basic. Okay, whoops, let me switch back to here. So that's histidine. Histidine is HIS, and an H, it makes perfectly logical sense. There's the backbone. Remember the linking carbon. Let me make sure I haven't skipped any along the way. Okay. Then it's the other one that reminds me of home plate for baseball. So this is what it looks like. Which for your functional groups, it's an imidazole functional group. So now the nitrogens are on the sides of home plate. Don't forget to do double bonds. Many times it is positively charged. Not always, but many times it can be because of that double bond. The pKa for it's perturbed to where we can have a hydrogen hang off of here, and it's a possible at times to have a hydrogen hang off of here because this is aromatic, and so then the positive charge is shared overall. Okay, even though a lot of times I'll show it on one nitrogen, it's actually shared because that double bond can swing from side to side. So it's aromatic, it's basic, which means it can be positively charged. It's got special pKa's. What else? Does anyone know what else it's really good for? So now I'm going to draw it where it's with the hydrogens on. Whoops. There's that hydrogen. And when the hydrogen's on this side, it's positively charged. Okay. But what's so special about it is we have, many times it acts as both a general acid and a general base. And it can do so in the same mechanism. Because we've got two sides, those two nitrogens, one can donate a hydrogen while the other one accepts a hydrogen from something else, and vice versa, so it's reversible. And this double bond can literally flip from one nitrogen to the other. So you can imagine something if, let me take off that hydrogen now. I didn't realize there were so many steps back. So we can imagine that we have something coming here and swiping that hydrogen. These electrons then, it doesn't want to be negatively charged. It can go somewhere, and it can go down here to this carbon, boom, to reform a double bond. This double bond that can go somewhere, and it can swing out to take or to attack something, and then it just goes backwards. So that's why it's both a general acid and a general base within the same mechanism. And so histidines a lot of times are used for what's called the catalytic triad, where you have another amino acid on one side, another amino acid on the other side, and they just kind of react with each other to make whatever be more a stronger nucleophile in the end, ultimately. And so you'll see histidines a lot. Okay, so we're doing time. So I'm trying to take time to go through all the 20 amino acids, which is so important. Yeah.
How can it be both a general acid and general base if it's basic? Like, I don't know. What do you mean by that?
When we say it's basic, it just means it has to do with its pKa.
Okay.
It's a general acid. So in that sense, here, let me show you. I'll show you a very specific example. So if you will indulge me, which I did not realize that hydrogen did not go with it. So ignore that, like the Wizard of Oz, ignore the man behind the curtain, ignore that hydrogen. So, okay. So we have this hydrogen. let's say, and this is a mechanism that you're going to see later on. There's another amino acid, I won't ruin it by saying what, but there's one that's a carboxylic acid. It looks like this. What can happen here is this can come out and reach and take this hydrogen. So in doing so, that is now a general acid. The histidine is a general acid. It's giving up the proton. These electrons can then come down here, boom, sound effects optional. These electrons here can swing out and take the hydrogen from something else, so therefore it's a general base. And so, for example, let's say that it takes the hydrogen from another one that we haven't seen yet, but we will. It looks like this. By taking that hydrogen, by taking this hydrogen away, it makes this oxygen a good nucleophile to go out and attack things. That's why that's called a chalic triad. This one's called an Histidine, which we'll find out later on. Chalic triad. It makes that thing, what is that blue? The oxygen in blue would be a really good nucleophile to go out and smack things. And then it's reversible. Because then whenever you're done with that reaction, you just do the reverse. And so the oxygen leaves whatever it had attacked before, takes the hydrogen back from the nitrogen on the right, which then the double bond swings the other way, and then the hydrogen goes back to the carboxylic acid. So that's why it's very reversible. And I wouldn't be surprised if some of your mechanisms for the enzyme that you choose have histidines in it, because histidines are really, really common. Okay, going along that line, another one, so histidine is a basic amino acid. I'm just going to continue on with the basic amino acids. One that you're probably going to almost get sick of seeing next semester, that's a basic amino acid, is called lysine. Okay, lysine's three-letter code is LYS, but its one-letter code can't be an L because L's are already taken. So it's a K. Like, it makes no sense. I just have to memorize it. Can't be Y because Y is taken. It can't be S, I, N, or E. All right, so every single letter is taken in lysine, so it's a K. Lysine's another one that people mess up on a lot of times. So this is the backbone. For its R group, what you have to remember is it's just an amino group. It has an amino group hanging off of it for its functional group, but the amino group is in that fifth position. It's called an epsilon amino group. So we have to go 1, 2, 3, 4. Make sure I didn't miscount. Then amino. And that nitrogen can be positively charged. That's the reason why it's a base. It can accept the hydrogen. Yeah, I'm trying to be neutral here. But what are amino, if you remember back to organic chemistry, what are amino groups amazing at being? Because the way I have it drawn here is it really looks like this. Nucleophiles. And good, it's also good leading group as well. But in fact, it forms what's called a shift base. It forms imines, which is one of the functional groups that you have to know from biochemistry, really, really well, and which is reversible. And so you'll see lysine a lot. And the other one that's the third of the basic amino acids, those are our bases. So we have histidine, we have lysine. Which yesterday was National Speak Like a Pirate Day. I heard that on the radio. Too bad we didn't have this lecture yesterday, because this one is the one that's the pirate amino acid. It's arginine. Whoops, I misspelled it. Arginine. And the reason why is because its three-letter code is ARG. Get it? National Speak Like a Pirate. Oh, come on, people. Grant me my nerdiness occasionally. All right, and this one letter code can't be A because this is alanine. So this one's R. R. Yeah. This was, I mentioned isoleucine used to give me fits. This one's another one. Drawing it used to drive me nuts because without fail, I would forget a carbon or something. But it's a guanidino group. So remember, there's the backbone. And what it is it's 1, 2, 3 total carbons, a nitrogen. Let me make sure I'm not assuming 1, 2, 3. Yes, I didn't. That just doesn't look right. A carbon, double bonded to a nitrogen, and has another nitrogen hanging off. And it essentially is almost always positively charged. It has a very, very high pKa. It can go into hydrogen. Just like this one can be positively charged, this one. Because of the fact, and there's resonance. I do it this way. That double bond could be at the bottom nitrogen, and the other one be a single bond. And so it's also great. It's great. All 3 of these, especially lysine and arginine, but histidine can as well, are great at forming what's called, the old term for it was a salt bridge, which means an electrostatic interaction. They're positively charged, so they can form a nice ionic interaction within the protein or between proteins with something that's negatively charged. Which I don't think we have enough time on today. On Friday, please write this down because I will forget. We're going to pick up with the negatively charged amino acids, and I'm going to do the polars. So those are the last 2 groups. Like I said, I'm going to try to take a lot of time to go through these, so that way you're ready for the quiz on Monday and everything. And on Friday, we're going to pick up with those that are negatively charged.